So, Watson and Crick, they mentioned in their paper that was published in 1953 that the specific pairing or complementary basis that is ATCG suggests a copying mechanism for the genetic material. So very early before they actually started investigating about the DNA replication, Watson and Crick highlighted in their paper that for this pairing to happen, this pairing happened because of suggested replication or copying mechanism, where each strand can serve to be the template, like the original, for the production of a new complementary, which is the copied strand, which was obvious by the base pairs that we know. And in their second paper, they focused more on the DNA replication, and they actually said the two words, or they defined the understanding of the two words: parent strand, which is the original DNA, and the daughter strand, which is basically the newly synthesized DNA strands. After some time, there was more into understanding what is replication. So there were 3 proposed methods for replication. These are hypotheses or proposed understandings. They're not 100% correct. And that was mid-1950s. The first conservative one was basically focusing about having our DNA molecule. Here, this is my parent, my old. You can see here the blue is the old or parent and the red is the daughter strand. And as you can see here, copying will happen, replication will happen. After that, the 2 old strands will come back together and the 2 new synthesized strands will join together. So I'll end up with 2 DNA molecules, one totally made of the original DNA, and the new one is made of one old, one new. The new one is totally made of new genetic material. And they call this the conservative model. Our next model is basically the middle idea of it. So it's called the semi-conservative way. So as you can see here, I have my original original parent strand of my DNA, and then I'll have here the 2 red daughter strands. And as you can see, after replication takes place, one old strand will wind up with one new strand. So I will end up having 2 DNA molecules where each of these DNA molecules has one old strand and one new synthesized strand. So each DNA molecule has one old, which is the blue, and one new, which is the red. And that's called semi-conservative. Our last understanding or last model was the disruptive. And here it's basically as if I'm chopping my DNA molecule into small pieces, and then I have my nucleotides here, and then I build them. So I have one old nucleotide with one new nucleotide, one old and one new, one old and one new in the same strand. So I have my 2 strands basically like a mix and match of old and new strands. Okay, and that's called the disruptive model.